Hello all, welcome once again to E-class. I am Dr. Tejinder Kaur Sarna. Hope you are enjoying your online sessions. Today we will be talking about body composition. What is body composition? Body composition is a term used to describe the percentage of fat, bone, water and muscle in human body. Water and fat are two chemicals that vary at expense of each other. Measuring body composition in human beings is a typical response to identify or depict any deficiencies or excess of component that is known to be a causative health risk factor. There are direct, criterion and indirect methods to measure body composition, each having its own advantages and disadvantages. By the end of this module, you will be able to comprehend the term body composition, understand the different components of body composition, understand the direct and criterion method of body composition and different methods to assess the same. Human body is composed of water, fat, mineral, proteins. Body on the basis of composition is divided into two components, fat component and fat free component, body fat being most variable component of the body. Fat in all the vital organs and in bone marrow is termed as essential fat, which is necessary for human functions, while that stored in adipose tissue is termed as storage fat. Let us see this equation. Human is equal to Cm plus ECT plus fat. In this expression, human comprises of cell mass, which is Cm, extracellular mass, which is ECT, and fat, which is FAT. While cell mass is a sum of actively mobilizing body cells, the extracellular tissue acts as a support for metabolically active cell mass. And fat, the third component, is the energy reserve held in the adipose tissue beneath the skin and around the vital organs. Sum of the cell mass and extracellular tissue is lean body mass, or it can be elaborated that the weight of muscles, bones, ligaments, tendons and all the internal organs combine to form lean body mass. Assessment of body composition is concerned chiefly of lean body mass around the percentage of fat and fat free mass in the body. Lean body mass and fat mass are often used interchangeably, but lean body mass includes essential fat while fat free mass does not, and approximately 70% of fat free mass is water based. Bone mineral mass is estimated by the mineral content of bone density. It constitutes of 50% of mineral and protein and rest 50% water. Total bone weight is approximately 12 to 15% of total body weight, but only 3 to 2% of this is mineral. Percentage body fat, which is dignified as percentage BF, is a percentage of body fat. Now that we have learned what are the basic components of human body, let us start with different methods of assessing the same. Methods of assessing body composition. Body composition can be quantified at 5 major levels: atomic level, molecular level, cellular level, tissue organ level and whole body level. At atomic level, human body is divided up to 11 components, major being oxygen, carbon, hydrogen and nitrogen, which constitutes of about 96% of body mass. At molecular level, human being is divided in 6 major components: water, protein, carbohydrate, lipid, soft tissue minerals and bone minerals. They can form different combinations in body to form FM, which is fat mass, fat free mass, lean body mass, which we have already discussed. Now these are gender, age based and specific to nutritional status also. At cellular level, body has three main components: extracellular fluids and cells. This can be further divided into fat, which is fat free mass, and cell, which is body cell mass. Fat and liquid are often interchangeable. The major components of tissue organ level includes adipose tissue, visceral mass, and skeletal muscles and bone. At whole body level, the composition is divided into three major parts: head, trunk and appendages, and these can be assessed by anthropometry circumferences at the very basic level. Whole body weight, volume, density, impedance can be assessed in body compositions. Now let us look at this table. This table gives a complete elaboration of the body mass, the components and the constituents. To assess the body composition, there are three different methods: direct, criterion and indirect methods, all having their own advantages and disadvantages. In this module, we will be discussing only the direct methods. Let us start one by one. Number 1, total body water. Total body water is an easy method to estimate the body composition at molecular, cellular and tissue level. The calculations are assumed based on the constant hydration of fat free mass. Since in all the adults total body water is almost constant, that is 73%, the formula becomes fat free mass is equal to TBW divided by 0.73. Total body water ration varies in certain nutritional and physiological conditions. In infants and children, total body water divided by fat free mass ratio is very different from malnourished patients, where with severe protein depletion hydration factors is as high as 75%. Under this technique, the first one is isotope dilution technique. Dilution methodology is based on the concept that certain substances are evenly distributed within specified fluid components, and that the dilution of a known component or a known amount of a substance introduced into an unknown volume permits the calculation of the unknown volume. Heavy water like titanium dioxide and titanium dioxide distribute throughout the body fluid, which makes it as a substance of choice for determining the total body water. Also known as hydrometry, this solution is used to measure total body water, therefore allowing fat free mass estimation. The calculated dose of titanium labeled water is given to the patient. After a set period of time, when the body has reached an equilibrium state, the enrichment of body water pool is measured using different samples like saliva, urine and blood. The advantage of this technique is its simplicity and minimum requirement. It has proved to be good for infants and toddlers and individuals with normal hydration. The only disadvantage is the hydration level of body that varies during certain physiological conditions, which can be overhydration or dehydration. Second technique is total body counting or total body potassium. This is also known as total body counting, which measures the amount of naturally present radioactive potassium 40 in the body. Once the total body potassium is known, fat free mass can be estimated assuming the normal or the constant concentration of potassium in fat free mass. Third technique is neutron activation. This is one of the most highly accurate measurement of tissue specific body composition, scanning the human body for 1 hour. The subjects are exposed to neutron field for a fixed time. Gamma output is measured as the skin muscle releases and goes back into pre-exposed state. This gamma output can be measured in two ways. Prompt gamma neutron activation, which is measured immediately upon activation, and delayed gamma neutron activation, which is measured after a delayed period of time. This technique can be helpful in measuring many elements of body like calcium, nitrogen, sodium, chloride. Body nitrogen can be quantified by this method. It also helps in predicting the amount of protein in the body, which further helps to analyze fat-free mass. These were the direct methods of assessing body composition. There are certain cation methods too. Let's discuss them one by one. The first one is body density, which is also known as densometry or hydrodensometry. This is the oldest means of determining the body composition, which works on Archimedes' principle, which states that when body is submerged in water, there is a Bion's counter force equivalent to the weight of the water which is displaced, as the bone and muscle are denser than water. The formula applied is body density is equal to body weight in the air divided by body weight in air minus body weight under water. The advantage of this method is that it gives the accurate results. The disadvantages are also there. The disadvantages is that this method, it is practically not possible for invalid children, old people, and at times people do not agree to get completely immersed in water. For this, we have another therapy, which is air displacement, popularly known as BOT-POD, is an alternate to the above techniques which uses pressure volume relationship to estimate body volume and body density. As you can see in the picture, the subject is made to settle down inside an egg-shaped pod which measures the volume of air displaced, thus allowing the pod, which is a BOT-POD, to measure the overall density, and then the body fat percentage is calculated through equation. This becomes very easy and useful in cases of children and adults who have fear of water or holding breath underwater. Computed SX-rated tomography, CT scan as popularly known, is one of the latest and accurate technique for measuring body composition, assessment, whole body quantities of fat and lean tissue. You can look at this picture. Apart from imaging capacities, CT scan also distinguishes body tissues on the basis of signal attenuation through the high radiation technique. It is especially helpful in assessing non-adipose fat and fat infiltration of skeleton, muscle or liver tissue. This method is not useful for obese people. The another very well known technique is MRI, magnetic resonance imaging. MRI is another important imaging technique which helps in estimation of volume rather than the mass of adipose tissue. Through this technique, images are produced based on the absorption and emission of energy in the ratio frequency created in an electromagnetic spectrum. Regardless of using high quantity imaging techniques, MRI data cannot distinguish adipose tissue from another fat mass in the body, or fat mass depicted by MRI is a part of adipose tissue. Advantages of MRI over other technique is its capacity to estimate regional body composition, and it is more accurate and viable approach in an estimation of intra-abdominal adipose tissue, which is only the disadvantage of being too expensive and is available in limited area only. Let's talk about DEXA. It is the latest, most accurate, most expensive non-invasive method of assessing body composition and gives the whole body scan, which includes body density, body fat percentage, lean body mass, fat mass, and the entire distribution of fat and lean tissue in arms, legs and trunks. Now, this DEXA is considered as a gold standard technique for the diagnosis of human composition. DEXA works by passing two X-ray beams through the subjects and measuring the amount of X-ray absorbed by the tissue it passes through, of which one is of high intensity and other is low intensity. Now, this is an indication of density of tissue it passes through and greater the introduction in X-ray intensity. Advantages of gold standard technique are that it gives accurate, repeatable and most precise results, is user friendly for subjects as well as for operators. The disadvantages include are the concern of the amount of radiation, huge side scanning beds, upper weight limits of the subjects, which is not possible for a larger population study and large population studies as well as large size people. Let us summarize our module on direct methods of assessing body composition. Body composition and growth are two components of health for individual as well as the entire population. Body composition can be quantified at 5 major levels: atomic level, molecular level, cellular level, tissue organ level and whole body. Human body is composed of water, protein, mineral and fat. Body on the basis of composition can be divided into two main components, fat component and fat free component. Body fat being the most variable constituent of the body. Water and fat are the two chief components of the body composition and they both work at the expense of each other.